Bismillahirrahmanirrahim, students. Our today's topic is actually the diagrammatic representation of different replication cycles of viruses. You have already studied that viruses are of different types. We like DNA viruses, positive sense RNA viruses, negative sense viruses. Those viruses which contain double-stranded RNA, and then last the special family of retroviruses. So there is a slight difference between the replication cycles of these all categories. So we will start from the replication cycle of DNA viruses. We will cover all four cycles in our today's lecture. In first step, you have already studied that bacteriophage virus contains DNA as a genetic material in the virus. So DNA of virus contain all type of information regarding the formation of proteins of the virus, and all information related to the replication of the DNA. So we have already studied that DNA viruses are mostly double stranded in nature. A few exceptions are always there, but majority of DNA viruses are have double stranded DNA in it. In You have already studied you have also already studied that after entry or after penetration and the after uncoating of viral DNA gets entered into the nucleus of the cell. So when DNA replicates inside the nucleus of the cells, then after replication, messenger RNA forms within the nucleolus of the cell. You should know about the some important things like RNA or ribosomes are responsible for the production of proteins and with the use of RNA. But the RNAs are actually formed within the nucleolus of the nucleus. In This actually is a basic phenomena and common phenomena. But those viruses which contain RNA and RNA replication enzymes as well, they can generate more RNA stands at outside of the nucleolus as well. So but in original cell, RNA production occurs inside the nucleolus of the nucleus. So for the when DNA so after uncoating of the viral protein, DNA gets entered into the nucleus of the cell, and when DNA enters into the nucleus of the cell, DNA gets start replication. Then after completion, after the formation of hundreds of copies of hundred of copies of DNA stands, many messenger RNAs formed from the expression of the genes present on the surface of replicated DNAs. It means it means hundreds of copies of viral DNA can express some genes which are responsible for the production of messenger RNA. Then messenger RNAs, by using the host cell ribosomes, form special proteins. These special proteins then use Golgi apparatus or endoplasmic reticulum of the host cell and form a structural protein of the virus. Then this is a major thing about this whole theory is that viruses replicates DNA of the virus replicates inside the nucleus of the host cell. Okay, after completion and after the formation of structural protein and the after engulfment of the DNA and complete when complete virus found viral cell gets ruptured and viral and hundred of virus released into the species or into the host out in the extracellular species of the host body. So actually this is the DNA. This is all about DNA virus replication. It's a simple phenomena. There is no any complex step or any additional step is involved in the DNA replication. So mostly eukaryotic cells contain nucleic material in the form of DNA. And most of the nucleic acid in the form of DNA present is inside the nucleus. So when DNA needs when DNA virus gets entered into the host cell, DNA of the host cell, DNA of the virus binds with the DNA of the host cell. So DNA host of the host cell is present inside the nucleus. So viral DNA enters inside the nucleus to for the attachment with the host DNA. So this is another important point. Positive sense RNA versus replication. There is a difference between the replication in few steps of the positive sense RNA versus replication and the negative sense RNA virus replication. In positive sense RNA virus replication, like in coronavirus replication, incubation period is actually is actually low, or incubation period of the virus is only for few days, consist of only for few days, and like in coronavirus cases, incubation period is 14 days. Within 14 days patient can present with sign and symptoms. What happens in the positive sense RNA virus replication? When after uncoating of virus, when RNA enters into the host cell, this RNA directly, I am saying, please note it down, that entered RNA of virus came in contact with the ribosomes of the host cell. So directly RNA from virus involves in the production of different proteins by binding with the ribosomes of the host cell. So आरएनए डायरेक्टली जो है वो राइबोसोम्स के साथ आकर अटैच होकर प्रोटीन फॉर्मेशन शुरू कर दी. सो आफ्टर आफ्टर अटैचमेंट फॉर एक तरफ प्रोटीन फॉर्मेशन कंप्लीट होगी, तो दूसरी तरफ क्या होगा, उसी की जो है उसी आरएनए को दैन फर्दर कॉपीज भी तैयार की जाएंगी. सो क्या हुआ, ट्रांसक्रिप्शन हुई, मींस प्रोटीन सिंथेसिस, एज वेल एज उसके बाद क्या हुआ था, आरएनए की रेप्लिकेशन हुई थी. So protein synthesis actually is a very major step in the RNA replication, and after the formation of hundreds of copies of RNA, many different many structural protein are formed in the at second last stage, and the assembly occurs. Assembly involves actually in the assembly is actually is the binding of nucleic acid, means RNA, and the structural protein of the virus to form a complete mature virus. This complete mature virus after completion of after completion of complete or mature virus, mature released into the host cell get burst, and the after bursting of host cell, hundreds of virus released into the extra cellular spaces of host environment. So here is first step is very important, because first step is actually major difference between positive sense RNA viruses and the negative sense RNA viruses. In first steps, translation starts directly from the RNA of the virus in positive sense RNA viruses. In case of negative sense RNA viruses, there is an additional step involved in the replication of RNA or transcription of RNA, because virus is actually hungry for the production, or hungry for many proteins. So for the production of protein, virus need messenger RNA. So negative sense RNA viruses, when uncoating of negative sense RNA viruses occurs, RNAs moves into the cytoplasm of the host cell. But this negative sense RNA has not the ability to directly bind with ribosomes of the host cell and protein formation. It means this RNA is negative sense. That's why this RNA of virus is unable to transcript immediately after entry. So what happens after that? After entry of negative sense RNA into host cell, first steps occurs is actually the formation of new RNA strand from entered RNA of virus. It means RNA replication occurs by using previous RNA strand. This is much important, because RNA of virus is not attacking or is not making contact directly with the ribosome of the host cell. First of all, new copies of RNAs formed. New copies of RNAs, when new copies of RNA formed, these new copies of RNA act as messenger RNA and come in contact with the ribosome of the host cells, and new proteins proteins of structural and non-structural proteins of the virus forms. On the other hand, this viral RNA also undergoes the replication as well. So negative sense and the positive sense RNA viral replication is very important, because in negative sense RNA viruses, there is an additional step involved in the formation, or involve before the formation of protein, which is actually formation of RNA from RNA strand. So formation of RNA from RNA strand is actually depend on one enzyme which is known as RNA dependent RNA polymerase. So negative sense RNA viruses contain a unique enzyme which is known as RNA dependent RNA polymerase. Okay. So I think we have discussed enough about negative sense RNA viruses as well. Here is another important family of viruses which is known as retroviruses family. Retroviruses family is actually a family of RNA viruses. So their genome is composed of with RNA, usually a single stranded RNA, but with positive polarity. It means the retroviruses contains positive sense RNA, but their mechanism of replication is different from the other positive sense RNA viruses. So let's discuss what happens in the in case of retroviruses after uncoating, after attachment, penetration, and uncoating of viral virus uncoating of virus. When viral genome enters into the cytoplasm of the host cell. This viral genome, as you can observe in step 3, this viral genome, which is actually RNA involves gets involved in the formation of DNA. I'm going to repeat it. After penetration or after uncoating of virus, when viral genome get virus RNA enters into the cytoplasm of the host cell, the DNA formation occurs from the RNA of the virus. It's a nature of virus that DNA formed from the RNA of a virus. So formation of RNA from DNA is known as transcription. So when formation of DNA occurs from the RNA, uh, this is happening in the third step of the viral replication. So formation of RNA from the formation of DNA from the RNA is actually reverse transcription. Transcription is the formation of RNA from DNA. But in case of virus replication, when a new strand of DNA is formed from the is forming from the RNA of the virus, this is known as reverse transcription, and the reverse transcription needs an enzyme, reverse transcriptase. So retroviruses family naturally contain an enzyme which is known as reverse transcriptase enzyme. वायरस में जो यूनिक चीज़ें हैं ना, जो यूनिक इंजाइम है, वो अपने साथ लेकर आता है, वो हमारे सेल में मौजूद नहीं होते. सो व्हेन DNA formed in the cytoplasm of the host cell, this DNA strand, that DNA strand enters into the host cell nucleus, because DNA of the host cell is inside the nucleus. So after formation of DNA strand from the RNA of the virus, this DNA strand enters into the nucleus of the host cell, and this DNA strand binds with the DNA strand of host cell host cell or and after binding with the host DNA, copies of DNA, so replication of DNA occurs. After the replication, number of hundreds of DNA strands of virus can observed inside the nucleus. So after formation of hundreds of DNA viruses, these DNA gets converted into RNA. So so when so all DNA convert into RNAs by the process of transcription, formation of RNA from DNA, and these RNAs are involved in the formation of these RNAs act as a also messenger RNA, so they are involved in the formation of different protein. So this is actually happening in the retroviruses cases. Okay, you can differentiate between few steps which are different in positive, negative, and positive RNA viruses, negative sense RNA viruses, and in case of retroviruses. So you can differentiate following steps. So be careful about all these steps in different viruses. So for the study and for the easiness of easiness of the for your easiness, I have just prepared this short presentation for you, ताकि आप लोग जो हैं इन डायग्राम से अच्छा समझ सकें. तो मेरी जितनी अच्छी कोशिश है मैंने आपको समझा दिया होगा होपफुली. सो अपना ख्याल रखें और जो है इस टॉपिक को ज़रूर एक दफ़ा पढ़ लें, और जो डिफरेंसेस हैं उसको अच्छी तरह याद रखें. यूएचएस में क्या क्वेश्चन आ सकता है? यूएचएस में किसी भी वायरस का जो साइकिल है वो पूछ सकता है. अगर वो जनरल साइकिल पूछता है तो आपकी बुक में डायग्राम मौजूद है, लेकिन अगर वो एक स्पेसिफिक जैसे पॉजिटिव सेंस या नेगेटिव सेंस या रेट्रोवायरस का पूछ रहा है तो यू शुड ड्रॉ स्पेसिफिक साइकिल. ओके, जो कॉमन होगा वो गलत कर दिया जाएगा. सो दिस इज ऑल फॉर द टुडे. टेक केयर.